Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Considerations for the Next Wave of COVID-19 Development. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will take approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Medpace, who developed the content for this presentation. Medpace is a scientifically driven global full-service clinical contract research organization, CRO. They provide phase 1 to 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Most recently, Medpace's infectious disease team has been actively working with the global regulatory authorities and sponsors responding to COVID-19. This aligns with the company's mission to accelerate the global development of safe and effective medical therapeutics through its high science discipline operating approach that leverages local regulatory and deep therapeutic expertise across all major areas. Now, it is my pleasure to pass over the controls to Jennifer, and Jennifer will introduce herself and the rest of the team that will be presenting here today for this webinar. So Jennifer, I'm going to release your mic. You may begin when you are ready.
Thank you, Sonia. Hello and welcome, everyone. We're pleased to have the opportunity to speak to you today about our experiences during the SARS-CoV-2 pandemic. Today's focus will be on the evolution of conducting clinical trials for COVID-19, and we'll be focusing on where we are and where we think that we're going to be going. My name is Jennifer Gelhar, and I'm a Vice President of Clinical Trial Management. I'm responsible for oversight of clinical trials in infectious disease, and I'm joined today by an expert panel that will be sharing their thoughts on the protocol design elements, endpoint considerations, as well as some operational considerations for running COVID-19 clinical trials. So first, I'd like to introduce the panelists. I'll begin with our medical experts. And so first up is Dr. Anibal Kalmaji. Dr. Kalmaji is board certified in infectious diseases with 20 years experience in an anti-infective drug development on both the pharmaceutical and CRO side. He has more than 10 years of direct patient care experience in infectious diseases in hospital and ambulatory settings. His clinical research background in antimicrobials includes COVID-19, severe bacterial infections, as well as fungal infections in immunocompromised patients. Dr. Kalmaji is based in Buenos Aires, Argentina. Next up, we have Dr. Philip Cole. He is a board-certified ID physician, also in internal medicine. He brings nearly a decade of experience as a medical monitor of clinical trials, inclusive of anti-infective trials. These trials included populations of adult hospitalized patients and outpatients, and include indications in bacterial and viral respiratory infections such as COVID-19. He also has experience with bacterial skin and skin structure and bloodstream infections and invasive fungal diseases in the immunocompromised host. He has provided direct patient care, inclusive of management of respiratory viral infections in the academic and private practice settings. Next, we have Dr. Hervé Marin. He is an infectious disease physician trained in Paris, France, with almost 20 years of experience in clinical research in both the CRO and biotech environment. Hervé specializes in the development of new antimicrobials with a focus in viral infections. And last but not least, we have Dr. Brian Murphy. He's an infectious disease physician with over 18 years of running clinical trials. He's been with Medpace over 9 years, running global ID studies, and has been very active over the last few months with the ID team on our COVID-19 initiatives. Brian has been engaged with companies running COVID studies from preclinical to phase 3 and works closely with our sponsors, developing clinical development plans and regulatory strategies. Moving on to clinical operations, we're joined today by Ms. Carrie Shield. She is a senior director who's been with Medpace since 2006. She provides key guidance to our team of clinical trial managers in driving complex global studies across many indications with a specialty in infectious disease trials. She's also an integral part of our core COVID-19 leadership team. So before we begin, I'd like to take a moment and reflect on why we felt this webinar could be of interest. So as the pandemic took hold, there was a robust reaction by the industry to put forth tremendous effort in the fight against COVID-19. We saw countless companies begin reviewing their products and pipelines to understand if they had a potential answer to this emergency. They really sprinted into action to address this unmet need. And with that, I have to say we have witnessed some really great things happening very fast. Teams have navigated changes in timelines, different regulatory settings, moving from a very face-to-face atmosphere to virtual settings. We've also seen the increased use of electronic media, such as e-consents and other systems that we'll talk about a little bit later. But it's almost as if this emergency has really forced the industry into the next era. And I think as the initial wave or the sprint is starting to subside, we are entering a sort of marathon phase in which we must not only continue our efforts within the current environment, but prepare for the second wave of activity that's predicted to come and be able to be adaptable and flexible to the changing environment and restrictions and or leniency that we are seeing. And we're hopeful today that this webinar provides you some ideas and thoughts on how to manage this ever-changing landscape. So with that, I'd like to now hand it over to Dr. Murphy, who will provide just a short overview on some of the experiences we've had thus far. So Dr. Murphy?
Thanks, Jen, and hi again, everyone. This is Brian Murphy. Very excited to be with you and part of this panel today. So, as Jen mentioned, this novel coronavirus pandemic is really something that, of course, none of us have ever really seen. It's a real global medical threat with an urgent and unmet need for therapeutics that we just didn't have. So what you see on the screen then is a quick snapshot of some of the experience that we have to this point. But this is really changing as quickly as the pandemic has, and it's been super exciting to see and work with a group of pharma companies and biotechs that have really stepped up to bring some of these products to our patients. And I've been primarily impressed by the huge number of collaborations across this sector in the past few weeks. Regulatory, operational, even business transactions that before COVID, they may have really typically taken months of negotiation are now being signed off and finalized in just a matter of a few days to maybe a couple of weeks. And that's a trend that really has not shown many signs of slowing. So as a result, and as you know, by the good show that we have on the call today, clinical studies against SARS-CoV-2 and COVID-19 have exploded over the last few weeks. Just yesterday, I went on to clinicaltrials.gov, and there are over 2,000 studies now in COVID-19, and on the WHO database, over 3,400. And that number is continuing to grow on a daily basis, so it may already be outdated. And these studies are moving forward. Obviously, there's an unmet need, and they're being supported by a flood of funding. Lots of biotech venture money that's there. Quick estimate, I looked at the first quarter of 2020, $5 billion has been committed to new VC funds focused on biopharm. Now, that's not all, of course, COVID-19, but a good part of that is being driven by the COVID pandemic, and that's just for the first quarter. And I don't think this is likely just a fast burn through money. In addition to private money, there's billions in non-dilutive money that's been available for some of these companies. In the U.S., in particular, BARDA and NIH are really leading the charge to hand out billions of dollars in emergency funding. And this is something that's expected to continue over the next several years, which really will allow for more planning and longer term research. So with this pandemic, we've all been programmed to move and And I think a theme that you'll hear in this entire conversation on the panel today is to rush in a way and on a scale that we haven't experienced before. And I know as we've engaged many of you, that's this feeling of urgency that we all know is really important, because we don't know how the pandemic is going to evolve over the next coming months or years. So early on, there were some pharmaceutical companies and biotechs that could take advantage of that initial rush, and they were looking to move products that already had initial approval and other indications and move them quickly into COVID-19, and that was really important. And they shared they perhaps were able to do this because of a mechanism of action that aligned with our understanding, or at least our understanding at that point in time of the disease. Many of these were antiviral or immune modulating type of drugs. You can see some of the mechanisms of action on the slide. And then there were others quickly followed with products that maybe didn't have approval for an indication, but they could pivot quickly into COVID-19 by leveraging some of the existing INDs and IMPDs into COVID. So now we're seeing more products that are being specifically designed and developed for SARS-CoV-2 and COVID-19. So just a snapshot, I won't go through all the details here to show you the breadth that the panelists will be coming and sharing those experiences with you over the next several minutes here. Jen?
Thanks, Brian. Oops, sorry. Okay. So then first up, I'm going to pull back Brian into the conversation, but I'd like to ask Drs. Murphy and Mamja Marin to describe some of their experiences running the COVID-19 trials during the pandemic, the elements of the designs that they've seen, as well as how you anticipate that these are going to change over time. So Dr. Murphy, I'll start with you.
Sure, thanks, Jen. So I'll start with where we are and where we were in protocol design. And in many ways, when we start to talk about this reflects this rush that I've already mentioned and talked about. But with all this rush, I think one of the key elements that we needed and still need to ensure is that decisions are being made, of course, very quickly, but also very thoughtfully. And as we've experienced, the regulatory agencies are clearly on board with that idea, moving quickly, but not necessarily taking risky shortcuts. And I think all of us that are in drug development understand the importance of that. So for example, when we start talking about design, we need to have a good understanding of a widely acceptable definition of a case. What is actually a COVID case? And I'll kind of start in the back of this and then hand it over to Hervé to kind of get his opinion on where we are going. So some of the earlier treatment trials, when there were fewer approved and available tests that would allow patients who were suspected to have COVID to enter a study, we just did not have those tests available. So many of these would then define a case based on exposure history and symptoms, and those cases were defined as suspected, probable, or confirmed cases. And just as I mentioned, this was necessary, due to the difficulties of obtaining the test in many regions. But PCR tests were eventually approved, and now many of them are more readily available, along with some serology tests to confirm a past infection, and even more recently, antigen assays. The suspected or probable case definition is really not as justifiable, I don't think. So for treatment trials, sponsors are now really documenting the diagnosis of COVID-19 as a laboratory confirmed disease, as in most cases, local or central PCR testing or antigen testing can be obtained. And just a quick note, when we're talking about testing this in general, antigen testing is one of the newest ones approved, and it is faster. So it's attractive in a lot of ways to get those results very, very, very quick. Perhaps it's got a little slower sensitivity, and since the first one was only cleared in the U.S. by the FDA in early May, I don't think they're as widely as available as PCR. So by and large, right now, we are still seeing patients diagnosed with COVID-19 through PCR techniques. And now we have updated guidance that addresses a large range of populations that are being studied in clinical trials. Of course, there are outpatient studies, inpatients on mechanical ventilation, and even exposed or at-risk healthcare workers. And we'll talk about some of that in just a bit. I want to step back just for a moment and look at, there are companies now that have planned on initial phase 1 programs. they've come and are developing drugs thinking in a very traditional format that they would have a single ascending and multiple ascending dose studies, SAD and a MAD, in a normal healthy volunteers. And interesting, many of these, or at least a significant number of them, are getting regulatory feedback that they should actually move their products straight away, not into normal healthy volunteers, but into COVID-19 patients. So understanding the case definition for your program is an important aspect really early on. And of course, there are prevention trials. Many of these are being conducted in communities with documentation of circulating SARS-CoV-2. And they can be either a pre-exposure prophylaxis trial, and in those cases are defined by people at high risk for SARS-CoV-2 with no symptoms. That's perhaps a healthcare worker or a first responder. And then there's post-exposure prophylaxis trial, where cases are defined, for example, maybe by a healthcare worker or a household contact that has no symptoms, but they have had documented exposure to a definite or clinical presumed case. Looking at baseline severity, I think, is something that's been in the updated guidance, and sponsors should really consider how baseline severity is in the definition of the case. There are, firstly, patients who are SARS-CoV-2 diagnosed by PCR positive, but they have no symptoms, so without symptoms, an asymptomatic patient. I won't get into the asymptomatic, pre-symptomatic definition here, but we'll say those patients are without symptoms. Next, a mild COVID-19 patient. These are, again, PCR positive or antigen test positive. Their symptoms are mild COVID-19. This could include fever in some cases, cough, sore throat, anosmia that many are aware of. But the key is there's no shortness of breath, so without shortness of breath there. A moderate COVID-19, again, is antigen or PCR positive. These patients have mild illness. Now there is shortness of breath, perhaps with exertion. And then the clinical signs of COVID-19, including perhaps some mild tachypnea or tachycardia, but oxygen saturations are still staying greater than 93%. These patients are on room air. We move on for a case definition of severe COVID-19. Again, positive by testing, moderate illness, shortness of breath or respiratory distress, more severe tachypnea and tachycardia. Saturations are now less than 93% on room air. And finally, critical COVID-19. I mean, these are the patients with respiratory failure and have a clinical diagnosis of respiratory failure. Whether or not they're mechanically ventilated is not necessarily the issue. There may be a resource limitations in terms of mechanical ventilation, so those may not be available. And there are moves now not to intubate these patients and manage them in very non-invasive ways with positive pressure ventilation, high flow nasal cannula. But these are critical patients. Some of them may be on ECMO, some of them may be in shock on pressors, and perhaps there's multi-organ system failure. So that's a case definition of severity. One of the hallmarks of this infection of COVID-19, I think, as most of you are aware, is ARDS. And another way of looking at a case then is through that ARDS. I think primarily the studies that we've seen in the sponsors that are moving this forward do use the Berlin definition for a case of mild, moderate, or severe, and that, of course, is defined by a PaO2 to FiO2. So that's a bit of the perspective and some of the characteristics of a case in terms of where we are right now and a super high level of some of the current testing that's offered. I want to flip it over to Hervé and maybe to give us some insight on your opinion on where things may go.
Thank you, Brian. I think the new guidelines in terms of severity of cases is really helpful in helping us matching the mechanism of action of a drug versus what is the adequate patient population. In broad terms, immunomodulators may be most effective downstream of the infection, while as expected, and I think as RSV taught us, the earliest an antiviral can be applied, the greater efficacy we will see. One of the difficulty and one of the things we'll need to plan for in the future or be able to be flexible is changes in standard of care. As Brian alluded to, initially ventilation was sort of the panacea with invasive methods for patients with end-stage respiratory disease. For various reasons, including the fact that the hemodynamic of COVID-19 is not strictly similar to other ARDS syndrome, it does appear that mechanical ventilation is actually deleterious in some of those patients. So we have to be careful in patient selection and also endpoint. As Annabelle will allude later, is that it's really the last resort at this point, and everything else would be tried before somebody puts a tube into that patient. And that's something that applies specifically to ARDS, but also to a lot of other aspects of the disease. As we learn from it, we also need to be cognizant of that in the patient population we enroll, the inclusion criteria and the endpoint defined, and with specific attention to non-respiratory events as well, liver, kidney, coagulopathy, and other aspects. So what I think the definition of cases will remain relatively stable. They do evolve along the spectrum, and for regulatory agencies, especially if you have a product which safety profile is already known, it's clear that the benefit-risk balance will change depending on which stage of infection you're trying to address. The earlier you are, the safer the drug should be, while at later stages they might be willing to take a little more risk on board. The other aspect, and speaking about drugs that are already studied in other patient population, is that at least at the early stages, agencies will want to see PK in SARS-CoV-2 infected patients versus whatever other population you have, as obviously the metabolism of the drug may be different in those patients that have an hyperactive immune system. And we'll get into assays and endpoints a little later, but I think the evolving landscape is what we need to plan for. The definition of population is probably one that has now sort of solidified, and that's really helpful. But there's still a lot of flexibility that we need to write into the design and then monitor very carefully the data to make sure it remains adequate. Back at you, Brian.
Okay, thanks, Hervé. We'll just continue to move down the slide to study size, and of course, this is a critical piece not only to regulators, but for me, you and your budgets. The size of the study needs to be discussed with the regulatory agencies, and it's got to be in context of the study goals and where you are in development. Is it a proof of concept study? What phase is it? And are there adaptive elements that can be there? So, since we are unsure of the window of opportunity to efficiently conduct these clinical trials, there is understandably quite a need to move into the clinic quickly, and we get that, and I think regulators do as well. So this may translate in some cases to sponsors really feeling pressed to move out of animal models into patients or normal healthy volunteer studies as soon as they've satisfied IND and IMPD enabling preclinical studies. And in some situations, maybe the regulatory requirements are met, but there's still quite limited information that you may have supporting efficacy of the product through an animal model. So at that point, then it makes sense to start with a really small trial as a pilot or proof of concept study looking for a signal of benefit before starting into a larger or a pivotal trial. And maybe that's with a surrogate or a pharmacodynamic marker. There's no acceptable surrogate for progression of COVID-19 at the moment, or maybe there are some improvement in clinical outcome, and I think some others will talk about that in terms of endpoints later. So here in these cases, then the sample size may be quite small, definitely less than 100 patients, and it may or very well may be dose exploring. In other cases, there you may have really compelling preclinical, or in some cases even preliminary clinical evidence suggesting efficacy or some marker of efficacy. So in these cases, some sponsors have decided, along with their regulators, that it's best at that point to move directly into patients and run a study that does have statistical size that's significant and designed to provide more substantial evidence of efficacy and adequacy of safety. So given how quickly this pandemic has changed and how the standard of care or best available therapy has evolved during the course of some of these trials, many of these trials have been, and we're seeing quite a bit of adaptive elements that are coming in there, where they do incorporate prospectively planned criteria. Maybe these are to stop the trial for futility or hopefully for overwhelming efficacy, with the idea of in that case then of expanding from a proof of concept study to a very large confirmatory or pivotal trial. Other adaptations such as sample size readjustment is something we've often seen here. This is all laid out in a protocol a priori. There's oversight from usually an independent data monitoring committee, and as our statisticians will tell us, it's in a manner, of course, to ensure control of the type 1 error rate when you do those adaptive designs where you're looking at efficacy. So typically this is a sample size that increases as the study is amended to improve statistical power on the study result. And again, with the idea of looking to move that into a registration study fairly quickly. But in some cases, there's also been studies that have reduced the overall anticipated final sample size. And in at least some of these, this has been because of a reduced number of COVID-19 cases, or that particular population that's been defined by however they define their case in certain regions. Now, when you do have a sample size reduction in the number of subjects, you need to really discuss this with the regulatory agencies. And I think the agencies will expect to see that you've at least had some demonstration of the efforts to expand the study to additional sites, knowing that clearly there is the infection, the disease is continuing to spread. It's there, it just may not be in the region that you're looking at in the population to numbers to meet your timeline. So this again gets into cost and time as obvious factors that well may be impacted in this. More time for enrollment usually equals more money spent. So these have to be weighed against the potential risk of interpreting these results from studies that don't have or haven't met their enrollment goals and maybe haven't met statistical significance there. Just one last point on the sample size issue, and before I hand it over to Hervé again, there are rapid procedures and opportunity that we have now for scientific advice and regulatory comments. So we definitely recommend to stay in close communication with the regulatory agencies in this point. And I think there is an understanding from these agencies that there has been and there will be fluctuations in regions in the incidence of SARS-CoV-2 infections, and clearly there are events outside of an ongoing trial that will occur and may need to be considered in a revision of the trial design or amendment of the protocol. So, but when that happens, of course, these revisions need to be justified. They need to demonstrate good intentions, and they're well motivated, and they're not just trying to make that shortcut. That is a theme, not that our sponsors have ever done that, but that is a theme in terms of the regulatory agencies really holding true to their expectations and what's required to move these forward, maybe at a expedited rate, but nonetheless there are some assumptions that in terms of the quality of the data and the expectations that they will satisfy certain requirements are still there. Hervé, you want to offer some thoughts on where we're going next in this regard?
Well, I think the collaboration with regulatory agencies has been really critical in that aspect of things, but it comes at a price. And we're starting to see agencies saying, your 15 patient proof of concept open label study is not going to cut it. And so it's probably better at this stage to look at, what Brian was describing, an adaptive design that would move you from a dose escalation, probably with some sentinel core, depending on what your safety profile looks like, to what the next stage is, and adapt it into a larger trial. That will have a greater chance of getting on top of the pile or even being reviewed if you look at what the Italian and now the MHRA are putting through. So it's something to consider. Well, it with so many trials, it's also your advantage from the site perspective. If they can only enroll 2 or 3 patients, they may not favor your trial versus one where they could get more patients enrolled. So it's something to consider as you engage into development, and that will also have an impact on your return on investment. To be very honest, is that what value will have a non-comparative trial in terms of raising funds for the next stage, knowing that the infection in it by itself is evolving. So what is your contribution of the drug and what is changes in standard of care is going to be difficult to assert. So that's something definitively to consider in your sizing, and your choice of endpoint, which directly relates to that study side. Adaptive is a very broad term. I think what we are speaking here is the ability to continuously review data, reassess sample size, but potentially make some tweaking in your endpoint. And for that aspect, it's probably generally advisable to collect more clinical data than less. This is really what regulators want to look at, and you may have to move from one initial primary to a more composite endpoint, especially thinking of contribution of mortality. If your endpoint is time to clinical event, well, how do you treat the patient who would die? Are they consider that they had only 2 days in the hospital because they died on day 2? That's something you'll need to address in your protocol and the way you will handle your data. Back at you, Brian.
Okay, thanks, Hervé. And I know we're getting short on time here already, so we'll move forward in terms of comparators. And I think most of this will be covered under Hervé's where we're going next. But just quickly where we are, there's really still no widely available comparator that's fully approved for COVID-19 patients. And I think it's clear best available therapy may very well vary regionally depending on the severity of the patient. I think it's understood that the background standard of care should be the same in both treatment arms. Everyone would agree to that. And I think you've got to expect that there will be and could be more off-label use of a number of medications to a certain degree, that would be inevitable. Because, we're still working to really fully understand the COVID-19 disease process. So since that's the case, then there have been certain times discussions around single-arm trials of any investigative product. I think rarely such a single-arm trial would be appropriate, and even more rare that it would be accepted, as just as I mentioned, the disease natural history is still not completely known. So where we are now is really placebo-controlled trials are the most common and is really what's expected from many regulators at this point. Hervé, some thoughts on comparators?
So exactly, we are looking at optimized background therapy as your placebo, to use a term that was coined in the HIV days. Understanding that it depends on the mechanism of action of your drug. You're not going to allow an antiviral if what you're trying to demonstrate is that your drug has antiviral potency. But for later stages, addressing symptoms of the disease, you won't be able to preempt the use of convalescent plasma, supportive care, obviously, and potentially some antivirals where and when they are available. That's something to consider in your protocols. Depending on the pharmacokinetics of your drug, you take a good look at what interaction could be expected between your compound and those potential drugs, and monitor carefully what the standard of care is in the specific geography that you're at, noting that it's going to be really difficult to preempt physicians or patients from receiving what is currently considered the standard of care. But going for a non-comparative trial from both a safety perspective, noting that there are SARS-CoV-2 related safety events that we are discovering every day. You don't want those associated with your drug. But also from an efficacy perspective, you want to see incremental benefit versus the standard of care. Back at you, Jen.
Thank you very much. Thanks, guys, for those insights. I think some of the key takeaways that I noted down were really, as Brian said on a couple of occasions, that while we know that there's a huge rush and these studies are moving at an accelerated pace, we do have to remember that there are no shortcuts. Additionally, just looking at being flexible, making sure that flexibility is built into the protocol as it allows. We've got a changing pandemic, everything is moving and shifting. For example, how standard of care could change will also then require adaptations to the protocol potentially. So just being flexible and trying to get that in up front and be aware of it is important. And then, of course, working with the agencies to the best of your ability. They seem to be very happy to engage, and it's really encouraged that you reach out for advice sooner rather than later. So then moving into the next part of our discussion today, I'd like to ask Drs. Khalmaghi and Cole to describe their thoughts on appropriate endpoints for COVID-19 indication, how these have changed over time, and what we could potentially see in the future. So Dr. Khalmaghi, I'll start with you.
Thank you very much, Jen. So there is no doubt that a very important component of a successful clinical trial, in this case for COVID-19, is to select the right efficacy endpoints. So the FDA released their guidance for developing drugs and biological products for COVID-19 treatment of prevention on May 2020, so 3 weeks ago. In that document, the agency proposes some key recommendations to select proper endpoints in any drug development program for drugs to treat or prevent COVID-19, considering 3 main factors. One is the population under study, the other is the clinical setting. We are talking about outpatients, hospitalized patients, or ICU patients, and the baseline severity, disease severity criteria, as described very well earlier on by Dr. Murphy. All of these factors, population, clinical setting, and baseline severity, are closely related to each other. So one of the key message from this guidance is that the agency wants that say that the choice, the time frame, and the interpretation of endpoints may differ depending on the population evaluated in the trial. So they are emphasizing the importance to differentiate different populations. And another key message here is that early interaction with and collaboration with the regulatory agency to discuss the appropriate endpoints for the study is the paramount importance. for the sponsor and of course for the CRO. The identification of risk factors for progression of severe disease are useful for study design, defining the population to be included in specific studies, but also for patient stratification. Up to now, the identified risk factors for death or progression to severe illness are the age, the presence of underlying comorbidities, and biomarkers. And these findings are, you can see that are mirroring when you see series from China and compared with series from European countries and from US. So the differences are very small and they reflect the same. So an early US epidemiology survey from US suggests that the case fatality rate was highest in persons aged more than or equal to 85 years old, with a range between 10% and 27%, and was less than 1% for persons with ages between 0 and 54. So as you can see, the difference is huge. The presence of underlying comorbidities are clearly related with bad outcomes, and they are very well described. The presence of cardiovascular or respiratory diseases, diabetes mellitus, chronic kidney disease, an immunocompromised person. There are others, but these four are the most important. And the third risk factor for progression or to have severe illness is the presence of some biomarkers. So COVID-19 has been associated with many inflammatory changes, ischemic complication, coagulation disorders, and endothelitis. Systemic vasculitis and cytokine-mediated coagulation disorders are principal actors of multi-organ failure in patients with severe COVID-19, and several biomarkers have been evaluated to help to predict outcome. We can classify those biomarkers, identify and related with poor prognosis in hematological, for example, the lymphocyte count, neutrophil count, and the most important one, the ratio between neutrophil and lymphocyte count. Some are inflammatory biomarkers like C-reactive protein, and others are erythrocyte sedimentation rate and procalcitonin. Others are immunological, like the levels of interleukin-6, and others are biochemical, like D-dimer, troponin, creatinine kinase, and transaminases. Maybe some new laboratory biomarkers could be identified through the accurate analysis of multicentric case series. For example, homocysteine and angiotensin 2 are suggested as another or different potential biomarkers that could be helpful. So some examples of important clinical outcomes measures used in COVID-19 treatments trials are all-cause mortality, the presence of respiratory failure with a very strict definition, for example, the need for mechanical ventilation, ECMO, as mentioned before, Brian, non-invasive ventilation, the need for invasive mechanical ventilation, the need for intensive unit care level care based on clear definition, the need for hospitalization based on clear definition and specific clinical criteria. This is for less severe patients. And finally, the objective measures of sustained improvement. For example, the return to room air or baseline oxygen requirement. What are the most common and recommended endpoints used in trials in severe and or critical patients with COVID-19? The examples of appropriate endpoints could be the all-cause mortality at an appropriate time point, for example, at least 28 days. This is the recommended primary endpoint in phase 3 randomized clinical trials in patients hospitalized and or with severe critical disease. Other endpoints commonly used and usually defined as secondary are the proportion of patients alive and free of respiratory failure as an appropriate time point, the assessment of clinical status, and again at an appropriate time point. That clinical status usually is evaluated using an ordinal scale that incorporates multiple clinical outcomes of interest: death, mechanical ventilation, and those outcomes are ordered by their clinical importance. Many studies are using right now these ordinal scales, and if you can see in some paper that this is the most common endpoint utilized in current phase 3 randomized clinical trials. So 27% of all clinical trials, randomized clinical trials, are using these scales. And the last endpoint that we can mention here is the time to sustain recovery assessed over an appropriate duration. Other important recommendation from the FDA to be considered is the duration of the trial. A strong recommendation from the FDA to the sponsor is to address potential relapses in their endpoint definitions to ensure the adequate assessment of the durability of response. So the trial should be of sufficient duration to evaluate safety and effectiveness reliable. For example, a 4-week duration will likely be sufficient to capture most important outcomes, mortality, in a trial of mechanically ventilated patients. Longer duration would potentially be appropriate for trials of patients who are less ill at baseline and for trials of preventive treatment. In some cases, longer follow-up should be considered to assess safety. What about outpatient treatment trials? Usually, the patients included in these trials are suffering mild to moderate disease. The recommended endpoints, and probably the most important one, is the proportion of patients hospitalized by an appropriate time point. For example, patients admitted to the hospital for COVID-19 treatment from randomization to 7 days after randomization. The time to sustain clinical recovery assessed in a specific time frame is another endpoint. And other endpoints that we are facing they are used in this type of studies are the change from baseline in overall composite scores and also in individual COVID-19 related symptoms scores. And finally, the individual measures of quality of life. And for this purpose, the use of EPRO instruments is the paramount importance. Usually, they are captured using this kind of instruments. What about prevention trials? The primary endpoint for this trial should be the occurrence of laboratory confirmed SARS-CoV-2 infection with or without symptoms. Through a pre-specified time point. As Brian mentioned before, we have 2 different types of prevention trials using drugs. One is the post-exposure and the other is the pre-exposure. For post-exposure, for example, 1 dose of hydroxychloroquine is one study that is ongoing. The number of confirmed cases of 2 weeks after the contact is the primary endpoint. Other studies uses a prolonged prophylaxis, and the time point to identifying confirmed cases would be later. For pre-exposure, for example, in healthcare worker or patient under very clear exposition, the time point is defined according to the duration of the pre-exposure prophylaxis. That could be 60 days, 90 days, or even 6 months. Also, it's important to assess if COVID-19 is milder in persons receiving prophylaxis compared with persons not receiving prophylaxis. So collecting this clinical outcome data, for example, hospitalization and data on symptoms to support such analysis. One common question is if viral shedding can be used as an endpoint. And the answer is that in phase 2 treatment trials, a biological measure maybe is acceptable as a primary endpoint to support a phase 3 clinical endpoint study. However, the biologic endpoints are not appropriate as primary endpoints in phase 3 trial because there is no established predictive relationship between magnitude and timing of viral reductions and the extent of clinical benefit of how a patient feels, functions, or survives. So this is what we are right now in clinical endpoints following the FDA guidance. But what are the futures? So Philip, would you please comment something about what do you think about could be the next steps in regarding time points for this disease?
Yes, thank you, Anibal. So what I would say is looking into the future for endpoint considerations, beginning with regulatory agency perspectives, there's always a challenge trying to look into the future and to anticipate where the regulators are going. I usually come back to first principles first. They want to ensure safety of a drug, that's a mandate, certainly for the FDA, as well as efficacy. It's certainly not enough that a drug is safe, it has to work. And so the agencies are willing to consider alternative designs and endpoints. Early on in COVID-19, there were submissions made looking at easier and simpler, more sensitive endpoints. For example, time to improvement on the WHO 9-point ordinal scale or the NIAID's 8-point ordinal scale, looking for, say, a 2-degree measure of improvement. An example of that could be a patient who's hospitalized, moderate COVID-19 in the hospital on oxygen therapy, but not on high flow therapy, non-invasive ventilation, let alone ventilation. And you'd want to see a 2-point improvement of them. So that would be essentially off oxygen, ambulating, and discharged to home. May still have some limitations to activity, but that's their status performance. I anticipate that as the pandemic moves along, there are more clinical trials being run in COVID-19, that the agency is going to move more towards a little bit more traditional endpoints. So say your all-cause mortality, you're alive and respiratory compromise free at, say, day 28 for a small molecule therapy. Some of the cellular therapies, advanced therapies, cell products may require longer follow-up per agency recommendation. In terms of populations, I think we're going to see a greater degree of interest in populations of interest that may have been skipped over earlier on in COVID-19 research. And there is some mention in the guidance currently, but I think moving forward, there will be much more interest in fleshing out what are the performance characteristics of these drugs in patients who have renal impairment, knowing that we do see a significant number of patients, perhaps due to COVID-19 directly, due to running patients dry, who are septic to prevent volume overload and minimize the probability of going on mechanical ventilation, and some other ancillary measures done for these patients that you're going to see an interest in stratifying by how ill they are and what type of therapy that they receive. Other items in terms of populations will also be certainly more interest in pediatrics, or for example, interest in women who are pregnant or possibly becoming pregnant. As data fleshes out further in the pandemic, that will become greater of interest. I think endpoints, as were mentioned, the agency has a great deal of flexibility, and so I would encourage you to think big, but engage in conversations early with them. I think both Anibal and Brian touched on earlier that to be thoughtful and but deliberative and your endpoints. And just an example, Sumati Nambiar about 2 years ago at AAA in Lisbon had mentioned that a biomarker doesn't have to be validated by the agency to be considered for a trial as an endpoint, thinking in the future for endpoints. That being said, if this is not a validated biomarker for the FDA, you should have that conversation with the division early, and they in turn will have a discussion with their diagnostics colleagues to ensure that everything is aligned and appropriate. So don't be afraid to think outside the box. Don't be afraid to engage in early discussion and really collaborate with your regulators. They want to ensure not only the safety of the population, but also their commitment that we're providing effective drugs in a timely manner as is feasible and safe. I would encourage you to think about whether it's endpoints or assessments on your studies, the keys of minimizing variability. We've seen creativity, Jen touched on this earlier, in terms of electronic informed consent, using telemedicine, interviews to assess patients via the phone, say for an all-cause mortality assessment, or whether or not a patient required hospitalization. And thinking about not requiring a clinic visit just for these, but also the possibility of a patient being able to be assessed remotely by phone. That being said, keep the method of assessment similar. So if it's a teleconference, do it by phone. If it's by video conference, do it by video. So you're not leading or creating undue variability in how you make those assessments. And I think it was touched on earlier, and pretty much by everybody, I go back to, again, first principles: ensure patient safety, ensure that their right of privacy is protected in terms of any kind of video or remote assessments that use the internet as a mechanism of collecting that information, as well as the overall integrity of the data and the ability to have that data either remotely verified initially as COVID-19 prohibitions and travel limitations are lifted by on-site monitoring. And with that, and keeping in mind of the time, I'm going to pass things back to Jen. Over to you.
Thanks, Philip. And I know we're getting very close to the top of the hour, which doesn't leave much time for the operational considerations, but just a few points from my side, perhaps, before I hand it over to Carrie to give an update on where we're moving forward at this point. Really what we've seen over time, and as the pandemic unfolded, is that sites were really reaching out to us in terms of wanting to be included on any potential trial that we would have or that would come our way. And the shift that we've seen certainly now is that the sites, they are, it's a saturated environment, and they are starting to have these COVID clinical trial committees where, in fact, they are the ones that are reviewing each opportunity and being very selective as to what the site will undertake. So as we move forward in terms of really trying to match the right sites, it's not only the capabilities in terms of startup, interest, et cetera, but it's also making sure you understand the setup at the sites in terms of how they're potentially using COVID committees to determine whether or not they'll participate. So really, the CROs and sponsors have been challenged with differentiating their studies and making sure the scientific interest is there so that they can successfully move them through the committee internally. I think some of the other things that have had to occur, at least from the CRO side, are certainly cross-functional meetings and really trying to decrease the standard timelines for system builds for everything else. And I think the teams have really responded great to that and are able to really get these studies up and running very rapidly. I think additionally, we've seen that the DSMB members and the teams that we've worked with, they've been very engaged. They're also wanting to do their part. And so we've seen a great commitment to make sure that those are all being built in parallel and so that we can have everything ready for the first patient, first visit. So Carrie, I'll hand it over to you now to maybe give some of your insights in these last minutes so that at least you can talk a little bit about how things are moving in the future.
Sure, thanks, Jen. And Jen accurately summarized kind of what we're seeing now operationally. And as far as where we are headed, we're going to continue to see this space as an ever-evolving dynamic environment. Really reviewing trends and daily analytics allows us to navigate the competitive landscape. This consistent monitoring allows us to plan for the regional shifts and develop a robust strategy for our feasibility and site selection while still considering the regional hotspots and seasonality. I think we're going to continue to see differences across countries in the potential second wave. And so really a key from our standpoint is careful planning and having contingencies in place. Understanding and being mindful of the latest site restrictions through careful assessment of each of the site status will allow us to plan around and work through those restrictions to help the site continue to be able to recruit, as well as have our CRAs be able to monitor. Another aspect is centralized access and distribution of supplies and equipment will really streamline and ease the burden on the site. And of course, moving into the data and technology, I mean, data is valuable, but only if you have the proper technologies and processes, systems to leverage and reduce the development time and cost. So utilizing various digital engagement capabilities, ePro, EDC, site source portals, various platforms for telehealth and virtual monitoring for the CRAs. I think e-consent, which was mentioned previously, and then a consideration moving forward of connected health devices. So remote patient monitoring technologies to assess various data points and allow physicians to closely manage patient care is something that we are going to see as we continue to move forward. Next slide. To hit on some of those key takeaways, I think the team is really around the electronic systems, and we've mentioned timeline. In order to make sure that you're minimizing delays, really ensuring that the e-systems chosen have time for quality and validated builds is going to be important. And truly simplifying the system, yet capturing the critical and key data to protect the data endpoints is going to be key. Working at risk to avoid delays, so collaboratively moving as many of the elements in parallel and being prepared to quickly make updates as necessary. Site and patient support is going to be paramount and needs to be a top priority. We depend on them for successful execution of trials, so truly considering how they will experience the trial firsthand is critical. And in some aspects, in order to help, the sites are assessing their budget and considering new formats for compensation for the remote monitoring piece and all the aspects that go along with that. And then on the patient side, consideration for direct-to-patient shipment. We will continue to see that utilized more moving forward. As far as technology, it's really an exciting time to embrace all the various platforms that are coming out and being released, and to find the best way to leverage these systems to reduce burden on site staff and patients. We'll need to do continuous training and education and stay abreast of the latest regulatory guidelines, and then share that in the development of our protocols, as well as pass that down to the site and even on the patients for patient material. And another main thing is flexibility and really being able to adapt quickly, and a thoughtful monitoring strategy, utilizing risk-based approach, focusing on the critical data around endpoints, and leveraging our virtual options as mentioned. Jen?
Thanks, Carrie. So I think that comes to the end of the discussions for today. I'm not going to reiterate what's here on the slide. I think they've been spoken about quite a bit so far today. So with that, I will hand it over to Sonia.
Okay, well, thank you very much. We've come to the end of the portion of this webinar. If we couldn't attend to your questions, the team at Medpace will try to connect with you after this presentation. Now, if you could, sorry about that. And if you have any further questions, please direct them to the email address that's on your screen. And you see it there, info@medpace.com. So thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Hold on, I'm going to change it over so you can see the actual email address. Okay, and there it is. So that's the email address. If you have any further information or questions, please send it to info@medpace.com. So as I was saying before, you will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event at that link, and also share this link with your colleagues when they register for the recording as well. So I encourage you to do that. And get more information or get even more out of today's presentation by downloading the supporting materials under the Handouts tab, and there's 2 materials there for you to download. Now, please join us in thanking our speakers from Medpace for that very insightful presentation. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now.